Welcome to this deep dive into cell cycle kinetics, really the engine of how cells grow and divide.
Exactly. We've got quite a bit to unpack here.
We do. We're looking at different phases, how, radiosensitivity changes depending on the phase,
hmm
and also how we actually measure all this stuff.
Yeah, and it's not just academic, right? This ties directly into radiation oncology practice, how
Yeah,
we fractionate, why some tumors respond differently. It's all linked back to the cell cycle.
absolutely. So let's jump in. The basic structure, what are the main stages we need to know?
Okay, so for mammalian cells, we generally talk about 4 active phases and then a resting state.
Right.
You've got G1, which is gap 1, then S phase for synthesis,
Okay.
G2, that's gap 2, and M phase, which is mitosis.
The actual division.
The division, yeah.
Yeah.
And then there's G0, G0, which is a quiescent or resting phase outside the active cycle.
Got it. G1S, G2M, and sometimes G0. Maybe we can break down what happens in each, starting with G1.
Sure, so G1 follows mitosis. It's a period where the cell, well, it grows. It gets physically bigger.
Mm-hmm.
It's synthesizing RNA, making proteins, basically getting all the machinery ready for the next big step, which is copying its DNA. Lots of transcription factor activity here, too.
Okay, so it's bulking up and preparing. Then it moves into S phase.
Yeah.
Synthesis?
Exactly. S phase is all about DNA replication. Every chromosome is duplicated.
So you go from having one copy of each chromosome arm, the chromatid, to having 2 identical sister chromatids joined together.
Precisely. It's a critical phase to ensure genetic information is passed on correctly.
Makes sense. After all that copying, we hit G2, the second gap.
Right. G2 is more growth, more protein synthesis, but it's also a really important checkpoint phase.
Checkpoint.
Yeah, the cell basically does a final quality control. It checks, did DNA replication complete correctly? Is everything ready for division? Any necessary repairs happen here before committing to mitosis?
That sounds crucial. You don't want to divide with damaged DNA.
Definitely not. Which leads us right into M phase, mitosis.
The main event.
The main event. This is where the nucleus divides, prophase, metaphase, anaphase, telophase, separating those duplicated chromosomes.
Right.
And then that's typically followed by cytokinesis, where the whole cell splits into 2 daughter cells.
And you mentioned something key earlier about M phase and radiation.
Ah, yes. Critically important. Cells in M phase are generally the most radiosensitive. We'll definitely circle back to why.
Good to flag that. Okay, so after M, cells can go back into G1, or what about G0?
G0 is that resting state. The cell isn't actively preparing to divide. It's metabolically active, doing its job, but not cycling.
Can it stay there forever?
Some cells do, terminally differentiated ones. Others can stay there for a long time, but can be stimulated to re-enter G1 and start dividing again if needed.
And how about radiosensitivity for G0?
Generally very radioresistant, often even more resistant than cycling cells. They're sort of hunkered down.
Interesting contrast to M phase.
Okay.
And there's a term TC.
Right, TCC. That's simply the total cell cycle time, the time it takes to go from one mitosis to the next. So G1 plus S plus G2 plus M.
Got it. The full lab time.
Exactly.
Now, thinking about that lab time TC, is it the same for all cells? Like, does G1 always take the same amount of time?
Oh, definitely not. This is where it gets really interesting and variable. The order of phases is fixed, but the duration can differ quite a lot.
How much variation are we talking?
Well, let's look at the phases. M phase, mitosis, TM, that's usually pretty quick and consistent, maybe around an hour.
Go.
S phase, TS, where DNA is copied, that's fairly consistent too, maybe 6 to 8 hours in many mammalian cells.
6 to 8 hours. Still pretty consistent.
And G2, TG2, the gap before mitosis. Also relatively fixed, perhaps 3 to 4 hours.
So if M, S, and G2 are relatively constant,
Yeah.
the variation must come from?
G1. TG1 is the major variable. It can be super short, like an hour or less in rapidly dividing cells, or it can stretch out for many, many hours, even days in slower cycling cells.
Ah, so that's what drives the big differences in the total cycle time, TC.
Absolutely. Think about Chinese hamster ovary cells,
Mhm.
CHO cells. They cycle fast. Maybe TC is around 11 hours. Their G1 might be just an hour or so.
Okay, fast cycle, short G1.
Right. Now compare that to, say, HeLa cells, human cancer cells. Their TC might be more like 24 hours.
Wow, double.
Yeah. And that difference mostly comes from a much longer G1 phase, maybe 10 or 11 hours in those HeLa cells.
So G1 really sets the pace.
It really does. It's the most flexible part of the cycle. Maybe a mnemonic helps here, like go see grandma maybe. G1, SG2M. But remember, G1 is the one that's
No.
like, well, maybe grandma's visit takes a really long time sometimes.
Oh, okay, that works. G1 variability is key.
Key takeaway.
So now let's link this timing back to radiosensitivity. You mentioned M is the most sensitive. How does the rest of the cycle stack up?
Okay, so yeah, M phase is generally the most sensitive. Survival curves often have little or no shoulder. Very vulnerable. Next most sensitive is typically G2 phase. Remember that premitotic checkpoint. Damage here right before division is bad news.
Got it. M and G2 are the sensitive ones. What about G1?
G1 is kind of intermediate, but it can be variable itself. Especially in cells with a long G1, early G1 might be relatively resistant.
Okay.
But as the cell gets closer to S phase, late in G1, it tends to become more sensitive again. It's gearing up. In cells with a really short G1, it's often just somewhere in the middle, sensitivity-wise.
So M, most sensitive, G2 next, then G1 is intermediate and variable. What's left? S phase.
S phase, and this is generally the most radioresistant phase among actively cycling cells.
The most resistant. Why is that? Isn't it actively copying DNA? Seems like a vulnerable time.
You'd think so, but actually the process of replication provides a powerful repair tool. Specifically, late in S phase is the most resistant point in the whole active cycle.
Late S. Even more resistant than early S.
Yes. The thinking is that in late S, you have the newly synthesized sister chromatid right there, perfectly aligned. This allows for very efficient homologous recombination repair.
Ah, using the identical copy as a template to fix breaks accurately.
Exactly. It's like having the perfect blueprint right next to the damaged section. Survival curves for late S phase cells show the biggest shoulder, meaning they can absorb more sublethal damage before being killed.
Okay, maybe an analogy like
Like having a backup generator. Or maybe like repairing a damaged road section when you have the plans and all the materials right there versus trying to fix it from memory later. Late S has the plans right there.
I like that. Makes sense. So the order most sensitive to least sensitive for cycling cells is?
Generally. M, then G2, then G1 with variability, then early S, and finally late S is the most resistant.
And don't forget G0.
Right, G0, the resting cells are also very resistant, often put somewhere around or even more resistant than late S.
Okay, that sensitivity pattern is crucial. M and G2 sensitive, late S and G0 resistant.
That's the core concept, and it underpins so much of why fractionation works in radiotherapy.
Right, hitting cells at different points in their cycle over time. But how did researchers figure all this out? How do you measure, how long S phase actually is?
Great question. One of the classic foundational techniques is called the percent-labeled mitosis technique, or PLM.
Percent labeled mitosis. Okay.
It's a bit laborious, but it gives you the absolute durations of TC, TG1, Ts, TG2, and TM.
How does it work? What's the principle?
It uses a radioactive DNA precursor, usually tritiated thymidine, that's thymidine with radioactive hydrogen, 3-HTDR.
Okay, radioactive thymidine.
You expose a population of growing cells to this 3-HTDR for a very short time, like a flash label. Only the cells that happen to be in S phase at that moment will incorporate the radioactive thymidine into their newly made DNA.
So you've specifically tagged the S phase cells.
Exactly. Then you immediately wash away the radioactive stuff and add a whole lot of normal, non-radioactive thymidine. This chase stops any further incorporation of the label.
Got it. So only that initial cohort of S phase cells is labeled. What next?
Then you start sampling the cell population at regular time intervals. Every hour, say.
Okay.
You take these samples, put them on microscope slides, and perform autoradiography. Basically, you coat the slides with a photographic emulsion. The radiation from the tritium inside the labeled nuclei exposes silver grains in the emulsion directly above them.
Ah, so you can literally see which cells took up the label.
Precisely. Then you look under the microscope. You identify cells that are in mitosis, you can tell by the condensed chromosomes, and you count what percentage of those mitotic cells also have the silver grains over their nucleus, meaning they were part of that originally labeled S phase cohort.
That's the percent labeled mitosis.
That's the PLM value for that time point.
And how does plotting this percentage over time tell you the phase lengths?
Okay, imagine the graph. Percent labeled mitosis on the y-axis, time after the flash label on the x-axis. Initially, the percentage is 0 because the labeled S phase cells haven't reached mitosis yet. They have to finish S and go through G2 first.
Okay.
The time it takes for the first labeled cells to appear in mitosis for the curve to start rising, that time delay corresponds roughly to the length of G2, TG2.
Ah, the time gap before the labeled cells hit M phase.
Exactly. Then the curve rises as more of the labeled cohort enters M. It peaks, and then it falls as they exit M. The width of this first peak, say at the 50% height level, gives you an estimate of the duration of S phase Ts.
The time it took for the whole labeled S phase group to pass through mitosis.
Kind of, yeah. And the time it takes for the curve to rise or fall gives an indication of T at the duration of mitosis itself, though that's often harder to measure precisely this way.
Okay, so we get TG2 and Ts from the first waves. What about TC and TG1?
You wait longer, and you'll see a second wave of labeled mitosis as the daughter cells of the first labeled cohort go through the cycle again and reach mitosis. The time between corresponding points on the first and second peaks, like from the 50% rising point on the first peak to the 50% rising point on the second, that gives you the total cell cycle time, TEC.
The time for one full lap.
Right. And once you have TGTs, TG2, and an estimate for T, you can calculate TG1 simply by subtraction. TG1 equals TC, TS plus TG2 plus TM.
Wow, that's clever. A bit involved, but very clever.
It was foundational. But yeah, it's laborious. And real PLM curves aren't always perfectly sharp peaks.
Why not?
Because, as we discussed, there's natural variation in phase lengths within a cell population. So the cohort spreads out, the peaks get broader and lower over time as synchrony is lost, and the label gets diluted with each cell division.
Makes sense. So are there more modern, maybe faster techniques now?
Absolutely. Flow cytometry really revolutionized cell cycle analysis. It's much faster and gives you data on thousands, even millions of cells very quickly.
Flow cytometry. How does that work for cell cycle?
Well, the most basic way is single parameter flow cytometry, measuring just DNA content.
Okay.
You take your cells, fix them to preserve them, and stain the DNA with a fluorescent dye. Propydium iodide, or PI, is a common one. It slips between the DNA base pairs.
And the more DNA, the more dye binds, the more fluorescence.
Exactly. Fluorescence intensity is proportional to DNA content. Then you run these stained cells single file through a laser beam in the flow cytometer. Detectors measure the fluorescence emitted by each cell as it passes through.
Then what does the data look like?
You get a histogram, a graph showing the number of cells, y-axis, versus the fluorescence intensity, which represents DNA content, x-axis.
What do you see on the histogram?
Typically, you see two distinct peaks. The first, larger peak, is at a certain fluorescence level representing a normal amount of DNA2N. These are your G1 cells, and G0 cells, if they're present, as they also have tuck-in DNA.
Okay, the G1 GOB peak.
Then you see a second, usually smaller peak at exactly twice the fluorescence intensity. That's 4N DNA content. These are your cells in G2 or M phase, as they've duplicated their DNA but haven't divided yet.
G2M peak. What about S phase?
S phase cells are actively synthesizing DNA, so their DNA content is somewhere between 2N and 4N. On the histogram, they show up as the cells distributed in the region between the G1 peak and the G2M peak.
Ah, so it's not a peak, but a distribution.
Right. And by using computer software to analyze the areas under the peaks and the region in between, you can calculate the percentage of cells in G1, G0, S, and G2M at that moment in time. It's a snapshot of the cell cycle distribution. You can also spot aneuploidy, abnormal DNA content if peaks show up in weird places.
That's much faster than PLM for getting the percentages.
Mm-hmm.
But it doesn't give you the phase durations, right?
Correct. Standard single-parameter flow gives you distribution, not absolute timing like PLM. However, there's a more advanced version.
Okay.
Dual-parameter flow cytometry. This lets you measure DNA content and active DNA synthesis simultaneously.
How do you measure active synthesis?
Similar idea to PLM, but using non-radioactive analogs of thymidine. Molecules like bromodioxyuridine, BRDU, or iododeoxyuridine, IUDR.
BRDU, I've heard of that.
Right. You give the cells a short pulse of BRDU. Just like the radioactive thymidine, it only gets incorporated into the DNA of cells that are actively in S phase during that pulse.
Okay.
Then you fix the cells, you stain for total DNA content using something like PI, which gives, say, a red fluorescence signal.
Got the red signal for total DNA.
And then you use a specific antibody that recognizes the BRDU incorporated into the DNA. This antibody is tagged with a different fluorescent marker, maybe one that glows green, like FITC.
So now cells have potentially a red signal for total DNA and a green signal if they were synthesizing DNA during the pulse.
Exactly. You run these through the flow cytometer, measuring both red and green fluorescence for each cell.
What does that data look like? A 2D plot?
Yep, a two-dimensional scatter plot, usually DNA content red on the x-axis and BRDU incorporation green on the y-axis.
How do the phases separate out on that plot?
It's really clear. G1 cells have 2N DNA content, low red, and didn't incorporate BRDU, low green, so they cluster in the bottom left.
Okay.
G2R cells have 4N DNA, high red, but also didn't incorporate BRDU during the pulse because they'd finished S phase, low green. They cluster in the bottom right.
Makes sense. And S phase.
S phase cells were actively incorporating BRDU, high green, and their DNA content is increasing from 2-N to 4N, intermediate to high red, so they form this distinct arc or horseshoe shape connecting the G1 and G2M populations, but up high in the green axis.
Ah, that gives a much cleaner separation of S phase than the single parameter histogram.
Much cleaner. It gives a more accurate S-phase fraction. And you can do pulse chase experiments with BRDU too, tracking cohorts of cells through the cycle over time to estimate things like Ts and even TC or potential doubling time, TPOT, which is a topic for our next deep dive.
Very powerful. Can you physically separate these cells too?
Yes, many flow cytometers are also cell sorters. Based on the signal, say, you want just the G2M cells, high red, low green, the machine can physically deflect those specific cells into a separate collection tube.
Wow, so you can isolate pure populations based on cell cycle phase for other experiments.
Exactly. It's incredibly useful.
This has been great, really clarifying the techniques. Shall we maybe crystallize some of the main points? Clinical pearls time?
Let's do it. Clinical pearls.
Okay, pearl 1. Remember, G1 phase is the most variable in length. That's key for differences in cell cycle time.
Pearl 2. Radiosensitivity order, know this. Most sensitive to least in the active cycle. MG2 G1, early S, late S. And G0 is very resistant.
Pearl 3. Percent labeled mitosis, PLM, the classic radioactive method. It determines the absolute durations of G1, S, G2, and M.
Pearl 4. Single parameter flow cytometry, usually with PI staining, gives you the cell cycle distribution, the percentage of cells in G1, S, G2M based on DNA content.
In Pearl 5. Dual parameter flow, DNA plus beta or UDR, gives a more accurate S-phase fraction measurement and can be used to track progression and estimate potential doubling time, TPOT.
Excellent summary. Ready for a quick board blitz?
Let's hit. Board blitz. Question 1. Which phase of the mammalian cell cycle generally exhibits the most variability in its duration? Is it AM phase, BS phase, CG2 phase, or DG1 phase?
That has to be DG1 phase. We talked about how that's the main driver of TC variation.
Correct. Rationale. G1 length varies significantly between cell types and conditions. Okay, question 2. Which of the following cell cycle phases is typically the most T radiosensitive in mammalian cells? A, late S phase, B, early S phase, CG1 phase, or DM phase?
Most sensitive. That's M phase, mitosis, so D.
Correct again. Rationale. Mitotic cells with condensed chromosomes are most vulnerable. Question 3. The percent labeled mitosis technique using tritiated thymidine allows for the determination of A, only the S phase duration, TAS, only the total cell cycle time, TC. C, the proportion of cells in G0 phase, or D, the absolute durations of G1, S, G2, and M phases.
PLM gives you the whole picture, the timing. So D, absolute durations of the active phases.
Perfect. Rationale. PLM analysis tracks labeled cells through the cycle, allowing calculation of all active phase lengths. Final one, question 4. In a single parameter flow cytometry histogram measuring DNA content with cryptidium iodide, cells in the S phase are represented by A, the peak with second DNA content, B, the peak with 4N DNA content, C, the region between the 7 and 4N DNA content peaks, or D, a distinct peak separate from 10N and 4N.
S phase is synthesizing DNA, so their content is between 2N and 4N. That's C, the region between the peaks.
Exactly right. Rationale. S-phase cells have intermediate DNA content as they replicate. Fantastic.
Good questions. Really reinforce the key points.
So thinking clinically now, how does all this cell cycle knowledge, the sensitivity differences, the kinetics, how does it actually impact our clinical decisions in radiation oncology?
Oh, hugely. I mean, the whole basis of fractionation relies on these principles, exploiting the differential sensitivity.
The 4Rs. Reassortment or redistribution is one of them.
Exactly. Giving radiation in fractions allows cells that were in a resistant phase, like late S, during one treatment to cycle into a more sensitive phase, like G2 or M, for the next fraction. So we kill more tumor cells over time. It helps overcome that resistance issue. It does. And also, measuring proliferation kinetics, like using flow cytometry to estimate the S phase fraction or even calculate TPOT, the potential doubling time. That can give us an idea of how fast a tumor might be regrowing between fractions.
Which might influence the overall treatment time or fractionation schedule.
Potentially, yes. For very rapidly proliferating tumors, there might be an argument for accelerated fractionation to counteract that rapid regrowth. It's complex, but kinetics can inform those decisions. It helps predict treatment response.
And what about combining radiation with chemotherapy?
Cell cycle knowledge is crucial there too. Many chemotherapy drugs are cell cycle specific. They might only kill cells in S phase or block them at the G2M checkpoint. Understanding this helps us design combination therapies, figuring out the best timing and sequencing to maximize tumor kill while hopefully minimizing toxicity. We need to know if drugs will synergize with radiation, perhaps by arresting cells in a sensitive phase, or if they might interfere.
So it's fundamental to fractionation, predicting response, and combining therapies. It really is central.
Absolutely central, understanding the enemy's life cycle, if you will.
A great way to put it. So, to wrap up today, we've covered the cell cycle phases, G1, S, G2, M, and the resting G0.
We talked about the variability, especially in G1, controlling the overall cycle time.
We detailed the changing radiosensitivity, M and G2 being most sensitive, late S and G0 being most resistant. Crucial stuff.
And we looked at how we measure these things, the classic PLM technique for absolute durations.
And the modern workhorse, flow cytometry, single parameter for distribution, dual parameter with BRDU for more accurate S-phase analysis, and kinetics.
Yeah, it provides the foundation for understanding how radiation works at the cellular level and how we can optimize treatments.
Fantastic. And as you mentioned, next time we'll build right on this.
Yep. Our next deep dive will get into tumor kinetics more broadly, talking about TPOT, growth fraction, cell loss factor, how the whole tumor population behaves.
Looking forward to that. It connects directly to what we discussed today.
It really does. It's the next layer up.
Okay, well, that brings us to the end of this deep dive. Remember, you can head over to radonc.smartlearn.com for practice oral boards.
And please subscribe to Radiation Oncology Smart Review so you don't miss our next session on tumor kinetics.
Thanks so much for tuning in.